In this video, I'll talk briefly about transcription and then talk in more detail about translation and the evolution of translation. In a previous lecture, I talked about some of the differences in transcription of tRNAs, rRNAs, and mRNAs, and this is just a summary of those. So I want to talk about some of the specifics of these transcription events, and then afterwards talk about translation over here and the evolution of translation and the genetic code. Here is an evolutionary tree or phylogenetic tree of the various RNA polymerases involved in transcription. And this diagram down here shows some of the subunits. So these main units of RNA polymerase 1 and 2 are common to all of them, and some of the others are common also. And so that's how simply it started, with only 5 subunits in bacteria. But again, these main ones are doing exactly the same thing in the eukaryotes as they do in the bacteria. And then there was further elaboration in archaea, 11 subunits in euryarchaea, and 13 subunits in crenarchaea, and that number 13 is unique to the crenarchaea over there. And then there were further elaborations in this, adding more subunits to RNA pol 1, pol 2, and pol 3. And again, RNA pol 1 transcribes the main ribosomal RNA genes. Pol 2 transcribes mRNAs. And pol 3 transcribes tRNAs and other small RNAs. And again, these are all related evolutionarily. The main differences are in the different protein subunits that have been added over the billions of years of evolution. Here are the ribosomal RNA genes. And in bacteria and archaea, these are transcribed by RNA polymerase. And then in eukaryotic ribosomal RNA genes, these are transcribed by RNA polymerase 1. And so there's a 5' external transcribe spacer region that contains a promoter. Then the small subunit genes, internal transcribe spacers, and a 5.8S gene in eukaryotic genes, which actually corresponds to the 5' region of the large subunit in bacteria. And then there's the large subunit of ribosomal RNA. 5S is also there in bacteria. The 5S gene in eukaryotes can either be located here or in a different part of the genome, and it's transcribed by RNA polymerase 3. The large subunit of the ribosomal RNA becomes the major part of the large ribosome subunit, and the small subunit gene becomes the major part of the ribosomal small subunit. And then these go together in this green, red, or green, blue, and yellow things here. Those are the tRNAs, where the tRNAs fit. The one at the A site, the P site, and then the exit site. And when the ribosome is put together, these two pretty much go together. You're looking at the open faces of those. So you're essentially looking inside the ribosome. Because ribosomes are needed in very large quantities, and ribosomal RNA is needed in large quantities, it's transcribed at very high rates, higher than almost any other gene. So if you look in the electron micrograph or microscope, what you see is something like this, where it looks like a feathery structure. And what this is the gene being transcribed continuously by RNA polymerase molecules, pol 1, lined up along that gene. So starting here, they're getting longer and longer and longer until you finally finish out here. So the small subunit gene would be about here, the large subunit gene would be about there. Upstream from that is a region that contains repetitive elements that look something like the promoter region, and so it lines up the polymerase molecules here so that they can easily be shifted to the promoter when that site becomes open. Because of the packing of the RNA polymerases on the gene, then if you can, this molecule can polymerize 50 nucleotides per second, but if you have 100 polymerase molecules on this gene at a time, then it's polymerizing 5,000 nucleotides per second, so at a much higher rate when it packs the polymerase molecules on that gene. In bacteria, the ribosomal genes are dispersed on the chromosome. So most bacteria have between 2 and 10 copies. A few have 1 copies, and a few have more than 10 copies, but by and large 2 to 10. E. coli has 7 copies, and because it has 7 copies, this is sufficient to produce 20 to 40,000 ribosomal RNAs and thus ribosomes in the 20 minutes during the time it's undergoing cell division. And that's enough ribosomes to load up into both cells, so they would have between 40 and 80,000 ribosomes. That's enough for each cell to survive, each of the daughter cells. So here's the processing for bacteria and archaea. Here's the ribosomal RNA gene with the promoter, small subunit, spacer, and large subunit. Transcription by RNA polymerase. You get the long precursor ribosomal RNA. There's processing. The small subunit is incorporated into the ribosomal small subunit, and the large subunit winds up in the ribosomal large subunit, and then they're put together for a functional ribosome. The arrangement is somewhat different in eukaryotes in that you have a transcription unit and they're in tandem copies, so you can see one transcription unit followed by another, and each one of those has the promoter. You then have a primary transcript with the small subunit, the large subunit here, and the 5.8S gene, or our ribosomal RNA, which then are processed into the 18S or small subunit, the 5.8S, and the 28S or the large subunit of the ribosomal RNA. And then that's the 18S is incorporated into the small ribosomal subunit, and the 25S or large subunit and 5.8S are incorporated into the large ribosomal subunit. And together those make a functional ribosome. So in many cases, all the ribosomal genes are located in one spot at what's called the secondary constriction on a chromosome. The centromere is the primary constriction, and it really is a constriction. But the secondary constriction is misnamed because it's actually a region that's unfolded, so that even during mitosis, the ribosomal genes are being transcribed. So the chromosomes are actually looping out there, and the reason it looks constricted is they don't stain very well, so it looks like an unstained region. So as I said, these are in long tandem repeats. In humans, there's about 200 copies of tandem repeats, and in Vishafaba, there are between 250 and 23,000 of these repetitive units in one location on the chromosome. And they look like this. Again, there's a small subunit gene, the 5.8S gene, and the large subunit gene. There's the promoter, and then there are these repeats where RNA polymerase POL1 stacks up in preparation for transcription, starting from the promoter. So there's the DNA that's transcribed into RNA, usually a 35 to 45S piece that contains the entire region. And then that's processed into the small subunit, the large subunit, and the 5.8S RNA. And the small subunit becomes incorporated into the small ribosomal subunit, and the large subunit and 5S RNAs, or ribosomal RNA, are incorporated into the large ribosomal subunit. Again, 50 nucleotides per second times 100 times 100 copies means that you can produce about 500,000 polymerized nucleotides per second in this. And still humans need about 200 copies, Vishwafaba needs about 250 copies, Drosophila needs at least 100 copies of these genes in order to make enough ribosomes for each cell. And processing is a little more complex in a eukaryotic RNA. So here's the gene up here with a small subunit, large subunit, and 5.8S actually comes in a couple different forms. One is a longer form. So the first thing that happens is you get transcription of the 35S precursor, and then the initial clipping is of the promoter region down here, and then finally the entire 5' external transcribe spacer is cut off. And then there's clipping in the internal transcribe spacer 1 to form a 20S piece and a 27S piece, again in two different forms. One is a long form and one's a shorter form of the 5.8S. And then the 20S is further clipped into the 18S that then becomes incorporated into the small ribosomal subunit. These two are processed by clipping ITS2, the internal transcribe spacer 2, and then that's cut into the 25S or large subunit and the 5.8S, either the shorter form or the longer form. And those are incorporated into the large ribosomal subunit. Another difference between bacteria and archaea and eukaryotes is that in bacteria and archaea, the ribosomal RNA genes and the mRNA genes are being transcribed in the same compartment, that is, the cell. And so the ribosomal RNA is transcribed, assembled into the ribosomes, and those attached directly to the mRNAs for translation. In eukaryotes, there's a difference because you have the ribosomal RNA genes and the mRNA genes being transcribed in the same location, but translation occurs out in the cytosol. So both the mRNAs and the ribosomes have to be exported out into the cytosol, and then translation occurs out there for many of them. Some are also translated in the endoplasmic reticulum, so that's a different compartment. And then those proteins which need to act in the nucleus, such as the RNA polymerase, DNA polymerase, etc., have to be imported into the nucleus for their function. Although this is more complex, it also is one way that gene expression can be more closely controlled. So here's a diagram of the eukaryotes with a little more detail. Here's the nucleus, and this is the nucleolus here, kind of oversized, but just to show you where ribosomal RNA is being transcribed and ribosomes are being assembled. So out here in the other parts of the nucleus, you're getting transcription of mRNAs. In the nucleolus, you're getting transcription of most of the ribosomal RNAs except for the 5S ribosomal RNA. You then have ribosomal proteins coming from the cytosol and then being assembled into the complete ribosome. The ribosome large subunit and small subunit are exported out of the nucleus separately. And in fact, the large subunit is one of the largest things that can go through a nuclear pore, and it has to actually distort a little bit in order to get out of the nuclear pore. Those are assembled around an mRNA message, and then translation occurs out here in the cytosol or on the endoplasmic reticulum. And then ribosomal proteins are imported back into the nucleus and the nucleolus, where then they act to build up new ribosomes. Ribosomal RNAs and tRNAs are very ancient molecules. This is a small tRNA. They're about 70 to 80 nucleotides in length. Here's the 5' end going through this green part, which is the D domain. Then the anticodon domain here, and then the T domain or T loop over here, and then back up to the 3' end. And it's after this CCA, which is added on post-transcriptionally, that's where the amino acid is added to the tRNA. Over here is the three-dimensional view of it, and then there's the acceptor arm. The T domain and D domain come together in the tRNA, and then this is the anticodon loop, and right there is the anticodon, the three nucleotides that hybridize to the codon on the mRNA. Unlike ribosomal RNA and mRNA and other RNAs, the promoter region for these genes is actually within the gene. So here's a tRNA gene, and it has an A box and a B box that will attach to transcription factor 3C, and then transcription factor 3C then will attach to transcription factor 3B, and then RNA polymerase 3 can attach there and start transcription. For the 5S ribosomal RNA, which is also transcribed by RNA polymerase 3, there's this different box called the C box, and that attaches transcription factor 3A, which then has affinity for transcription factor 3C, and that attaches transcription 3B, transcription factor 3B, and RNA polymerase attaches to that, and then transcription begins. In bacteria, they don't have these transcription factors. They have sigma factors, which serve the same function. So in eukaryotes, again, this is for Saccharomyces cerevisiae yeast. It's actually a little more complex than the previous diagram showed, in that transcription factor 3C consists of 6 separate subunits, separate proteins. The 138 kilodalton protein attaches to B, the 95 kilodalton protein attaches to A, and that has affinity from the 131 kilodalton protein with the 70 kilodalton protein in transcription factor 3B that has this protein, which is known as a TATA binding protein. Now, TATA binding proteins are known in mRNA genes. There's a TATA site that's an AT-rich region that the TATA binding protein binds to. In tRNAs, there is no TATA site, but they still have the TATA binding site, indicating that it preceded actually the TATA site in the mRNA and was there probably a lot longer in the past, probably 4 billion years ago. Also, as you saw from that initial phylogram showing the different subunits for RNA polymerase 3, there are many subunits in RNA polymerase 3. So the entire complex just to transcribe tRNAs is actually very complex in eukaryotes. And this is a little artist rendering of those two. So here's the 5S ribosomal RNA that's getting ready to start transcription. There's TF3A, TF3C, TF3B with the TATA binding protein, and then there's the RNA polymerase 3, and there's the number 1 site that is the first nucleotide that's going to be transcribed. Down here with the tRNAs, here's the TF3C that attaches to the A and B sites, and then TFB or TF3B with the TATA binding protein and RNA polymerase 3, and there's the +1, the first nucleotide that will be transcribed. Some of the tRNAs don't have promoter regions, and instead they're produced as operons. That is, the initial tRNA has a promoter, and RNA polymerase will sit on that and start transcription. And then you get transcription of all, in this case, 6 tRNAs. And then there's processing down to about 300 nucleotides each, and then final processing and folding, making each of the tRNAs about 70 to 80 nucleotides in length. And here we have mRNA genes. This is a bacterial mRNA, so it would be transcribed by just RNA polymerase from bacteria. This is the lac operon. In bacteria, often you have a transcript that encodes for several different proteins, in this case 3, LAC-Z, LAC-Y, and LAC-A. And then there's a gene that's upstream from that, the LAC-I, which controls the expression of that operon. The LAC-I gene is transcribed into mRNA and then translated into the LAC-I protein, which is a repressor protein. So when the amount of lactose in a cell is low, the repressor can bind to the operator, and this then stops any transcription of the lac operon. However, when the concentration of lactose increases, it binds to the repressor protein and then won't allow it to attach or stick to the operator. So then that opens up the promoter region for transcription of the lac operon. So when transcription begins, it transcribes the entire region so that the mRNA has the codes to make the LAC-Z, the LAC-Y, and the LAC-A proteins. So at the beginning of the untranslated region, there's a Shine-Delgarno sequence, or an anti-Shine-Delgarno sequence, that attaches to the ribosomal RNA of the small subunit that loads this message into the ribosome, and then there's an AUG start site that starts translation in the ribosome. Then the entire message is decoded and produces the LAC-Z protein, and at the very end it has a stop codon sequence, which is UAA, 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 and which essentially says stop, stop, stop. And then further on, there's another Shine-Delgarno sequence, or an anti-Shine-Delgarno sequence, that attaches to the ribosomal RNA of the small subunit and starts translation of that second protein, or that second mRNA. There's an AUG start, and then eventually an UAA stop, so that produces then the LAC-Y protein. And then there's another Shine-Delgarno sequence. The start codon this time is a UUG, and that makes the LAC-A protein followed by a stop to make those 3 proteins. The LAC-Z protein is the beta-galactosidase that cleaves lactose, which is a disaccharide, into 2 individual sugar units. LAC-Y and LAC-A aid in importing lactose into the cell. Eukaryotic polymerase, POL2 in this case, for transcribing mRNA genes, contains not only the RNA polymerase, but also a number of other proteins that help RNA polymerase bind to the promoter region, including to the TATA box shown here. They often have also activator proteins attached to that, which are ones that attach to enhancers, upstream or downstream enhancers. They're usually up around 1 kilobase or so upstream or downstream from the gene. And that aids in aggregating all of the transcription factors in the RNA polymerase in the promoter region. There also are silencer regions that can bind a repressor, which then will inhibit the enhancers and activators from initiating a transcription complex. So here's a general picture of a eukaryotic mRNA gene. As I said, upstream, they can also be downstream. There can be enhancers, one or more of those. And then between 75 and 90 bases upstream from the promoter, the start of transcription, there's often a GC box and what's called a CAT box, and in this case it's CCAAT. And then about 25 nucleotides upstream from the transcription start site is the TATA box that binds with the TATA binding protein to position the RNA polymerase in the correct position to start transcription. So then there's a 5' untranslated region, an exon. There can be introns, 1, 2, or sometimes dozens of introns. And exons, again, dozens of those are possible. And then a 3' untranslated region. So those are all transcribed. A cap is added, the 7-methylguanosine in the 5' to 5' orientation. Splicing then occurs, a poly A tail is added, and then that mRNA is finally exported from the nucleus. And only after it's been exported from the nucleus can it be translated into a protein and either wind up within the endoplasmic reticulum or in the cytosol. Many genes, especially those in eukaryotes, have exons and introns. That is, the exons are the expressed portion of the RNAs and genes, and introns are the intervening sequences between the exons. Sometimes the introns actually contain genes themselves. So, for instance, the globin gene is about 1,525 base pairs in length, with 622 of those in the exons and 893 in the introns. And most of the time this is the case where the introns are actually larger than the exon portions. One of the largest ones is the one for dystrophin, the gene and protein that go bad in muscular dystrophy. And that has over 70 introns and exons, and the gene itself is over 2 million base pairs in length, and most of that is in introns, and very small proportions of that are exons. And mRNAs then are translated using a universal genetic code. And I have universal in quotes there because it's not completely universal. There are many exceptions to portions of the genetic code. And I'll talk more about the evolution of this translation system, but in general you can see that mainly nonpolar amino acids are on the left-hand side. The polar ones are interior to that and equally on the left and right-hand side. And then you have a few places where there are positively charged amino acids, and then there are negatively charged amino acids. So one thing to notice is that there is structure to the genetic code, indicating that its evolution was somewhat structured. But it's also very complex, indicating that the evolution was probably complex as well. One thing that is obvious is that it's much too complex to have originated as it is today, and that it likely started from a much simpler system. So I want to discuss in a little bit of detail the evolution of translation by ribosomes, and part of this is looking at the evolution of the ribosomes themselves. This diagram is a little complex, so I'll go through it in pieces, but essentially this traces the evolution of all of the RNAs that are involved in translation, including the tRNAs, mRNAs, and ribosomal RNAs. as well as two different enzymes. These are amino-acyl tRNA synthetases. These are the enzymes that attach the amino acid to the proper tRNA. So if we just start with tRNAs, the CCA, which is the attachment site for the amino acid, appears to be one of the first portions of the tRNA during evolution. And then the acceptor arm, which consisted of a longer piece of RNA, was added. This consisted of approximately 10 or 20 nucleotides. It then got longer by adding the T pseudo C arm, and then the D arm and anticodon arm were added when the duplication of the entire molecule occurred. And so it doubled in size at that time and took on the shape that it has today, that cross structure that you see in all tRNAs. So we've pretty much traversed a span from about 4.2 billion years to about 3.2 billion years. Then these variable loops are added on, and that increases the complexity and the number of different tRNAs. And finally, the tRNAs then become, each of the arms becomes removable, and you can replace arms back and forth, which also increased the diversity of those tRNAs. The mRNAs came a little later, and they were fairly simple originally, just consisting of a short strand of RNA. Then for a time, it appears that they were double-stranded, so that you could read the bases in one direction and get one set of amino acids added, and in the opposite direction, on the opposite strand, was a different code. And so at that point, you have ribosomes starting to get a little more complex too. This is between about 3.8 and 3.4 billion years ago. Because RNA cannot maintain a double-stranded structure for more than 4 to 7 nucleotides at a time, this double-stranded nature of those early mRNAs became impossible as the mRNAs became larger. So they became longer and single-stranded at that time. And then introns began to invade those mRNAs, especially spliceosomal introns, until today now you have a poly-A tail, and there is a cap, the 7-methylguanosine, and increased numbers of introns. The small and the large ribosomal subunits, before they became parts of the ribosome, were probably RNAs that performed different functions. In particular, the small subunit ribosomal RNA may have been a molecule that delivered RNAs to other parts of the organism. The large subunits early on gained the peptidyl transferase center, that is the enzymatic portion of the ribosome that puts together the amino acids via a peptide bond. So that's the enzymatic portion of the ribosome. Then they tended to get larger and larger, the small subunit primarily involved in delivering the mRNA to the large subunit, to the peptidyl transferase site, as well as binding to the tRNAs, and the large subunit also bound to those tRNAs. But the small subunit became a ratchet system too, for moving the mRNA through 3 nucleotides at a time, corresponding to the triplet codons in the mRNA. With each additional piece added after that, they became more discriminating as to when they would translate and what they would translate. So all of the other forms, the additions that happened after about 3.2 billion years ago, tended to make them more efficient and more discriminating as to when and where they would translate those mRNAs into proteins. The complete ribosomes look something like this, where in bacteria and archaea there are about 21 proteins and a small subunit ribosomal RNA in the small subunit. And in the large subunit, about 34 proteins and a large ribosomal subunit, as well as a 5S RNA. In eukaryotes, they're much larger, so about 33 proteins and a small subunit ribosomal RNA, which is larger than the small subunit ribosomal RNA in bacteria and archaea. And in the large subunit, 49 proteins, sometimes more, and a large ribosomal RNA subunit, a 5.8S RNA, and a 5S RNA in the large subunit. And again, the small subunit is primarily delivering the mRNA to the large subunit, has attachment sites for the tRNAs, and also ratchets the mRNA through the ribosome at 3 nucleotides at a time. The large subunit, on the other hand, has sites for the tRNAs. It also has the enzymatic portion, so it is a ribozyme. That is, it performs the task of making the peptide bonds between the amino acids, and it also has a pore through the center through which is thread the newly formed polypeptide. Ribosomes are in every living cell, and that implies that they evolved once in the group of organisms that we see today, including the bacteria, archaea, and eukarya, and that they were very successful. And they've become so complex now that a new molecule that does the same thing probably isn't possible. It would take billions of years or so to evolve a new type of ribosome. Because ribosomes are universal, then you can make phylogenetic trees based on their sequences, and this is one based on the small subunit ribosomal RNA. And so you can see there's the bacteria, all the groups there, the archaea, and then the eukaryotes over here. So there have been many such trees produced. You can make essentially a universal tree, or a tree of life, of all life. This is a view where the large and small subunit have been pulled apart, showing where they come together as the two subunits. And there's the A, P, and E sites for the tRNAs. The A is the amino, where the new amino acid comes in. The P is where the peptide being formed is held, and the E is the exit site where the tRNA, now without any amino acid, is ejected. And there's how the mRNA is threaded through the two subunits. So currently, translation works like this. There's the mRNA, the 5' end of the mRNA, and the ribosome travels in the 5' to 3' direction down that ribosome, or you can look at it the other way, where the mRNA is being pushed through the ribosome in the opposite direction. Either way is fine. So there's the EPA sites, or EP and A sites. So when there's an AUG start codon, the initial tRNA binds to the codon with its anticodon, carrying a methionine, usually. Not always, but usually. Then the ribosome moves down, being pushed by the small subunit by 3 nucleotides, and that moves that methionine codon with the tRNA over to the P site. And that opens up the A site for the next tRNA with its amino acid to be placed into that A site. And GTP is needed in that case. So GTP is needed each time a new amino acid is placed into the A site with its tRNA. So then the peptidyl transferase, the PT, binds those two amino acids together, and it releases the amino acid in the P site, and then that tRNA moves over to the E site, and now it doesn't have an amino acid. The dipeptide in this case that has now two amino acids attached to it and attached to the tRNA at the A site shifts over to the P site, and that leaves the A site open for the next amino acid and tRNA to pop into place. So this process continues, shifting the tRNAs with their amino acids and shifting the tRNA without its amino acid to the E site over and over and over again until a stop codon is reached. In that case, a release factor causes a stoppage of translation and a separation of the two ribosomal subunits, which also releases the newly made polypeptide or protein. Several research studies have shown that the ribosome did not start out as this large structure, and I showed that a few slides ago. And so it looks as if the protoribosome, with the peptidyl transferase activity already, started out just as one site. But this was very inefficient at putting polypeptides together. Sometime in the past, that central part was duplicated, and it consists of about 110 nucleotides. And now you had a P and an A site. So there was a P site to hold the growing polypeptide and an A site to hang on to the new tRNA and amino acid to be added. Later, there were more parts added, so now you have an A, P, and an E or exit site. And then additional pieces were added that would further add discrimination to the translation process and make it more efficient as well. So it's been likened to an onion that is growing from the inside. You have these internal, very well preserved sites, and going to the outside where you have layers of new RNAs being added to the major structure. And this is just a model for the large subunit. The small subunit also was evolving to attach to and assist the large subunit. These are models from one of those papers I was talking about, showing this protoribosome with the peptidyl transferase activity, and then the foundation for that mainly to hold it into its catalytic form, and further adding layer after layer to this in what's called the onion model. So on the left-hand side, these are the different parts that were added on during evolution of the ribosome, and the other part there is the small subunit, and no detail is shown for that one. And as I mentioned a few slides ago, the genetic code has been studied also as to its evolution, and it's clear that it started from a much simpler code, and I want to talk about that now. So if we have a bit more detail in the genetic code here, showing also the structure of the R group in each one of the amino acids, then if you go from the bottom of this genetic code to the top, you find that you have simpler molecules on the bottom, glycine being the simplest, just having a hydrogen, and then alanine having a methyl group, and valine having an ethyl group, etc. And then going up to the top where you have more ring structures and generally more complex molecules. Also from left to right, you see some differences that are mainly nonpolar on the left, and then polar up in the upper sort of upper 2/3. And then there are positively charged basic groups, arginine, histidine, and lysine, and then the negatively charged amino acids, including aspartate and glutamate. So the structure of the genetic code then tells you, or at least gives you some clues as to how it evolved. So here are the amino acids grouped as to what they do in a protein. So down here you have alanine, valine, isoleucine, leucine, methionine, etc., getting larger and larger and primarily having some carbon-hydrogen groups, some hydrocarbon groups coming off of them. And these form primarily alpha helices and beta sheets in the protein. Likewise, these uncharged polar amino acids here, serine, threonine, asparagine, etc., also aid in forming alpha helices and beta sheets. And then you have these charged groups up here, positively charged, including arginine, histidine, and lysine, and then the negatively charged ones, aspartic acid and glutamic acid. Now those then are reactive groups, so those would be in an enzyme somewhere in the active pocket of those enzymes usually. And then there are a couple of amino acids over here that perform specific functions. These are cysteines, and so those have a sulfhydryl group that can either reduce or be oxidized in certain proteins, and also form disulfide bridges to hold covalently hold the structure of the protein. This is selenocysteine. That's a special one that's in specific proteins, acts sort of like cysteine, but has special functions. Glycine has only hydrogen. That allows free rotation. It's pretty much a placeholder in the polypeptide chain of the proteins. And then proline it's an amino acid rather than an amino acid, and that forms rigid kinks in the protein structure, the protein chain. And if you look at those 10 amino acids that were available 4.2 billion years ago, proven by a couple of different pieces of evidence, including Stanley Miller's experiments and the Murchison meteorites. These 5 are on the bottom row of the genetic code, and 4 of these 5 are on the next row up. So that tells you something about the evolution of the genetic code already, that it probably started from the bottom of that code and worked upwards. Also, those are among some of the simplest amino acids, and of course they were available a long time ago. So here are those 10 amino acids: alanine, valine, isoleucine, and leucine, aspartate or aspartic acid, glutamate or glutamic acid, glycine, proline, serine, and threonine. So you have members of each one of these categories. These ones would be reactive. These ones would form certain structures, or actually glycine, it just allows free rotation. It's a placeholder. And then these two could help to form alpha helices and beta sheets to hold those reactive groups into the proper conformation in the protein. So already 4.2 to 4.1 billion years ago, there were sufficient amino acids around to make functional proteins and peptides. So on this bottom row, then these are 5 of the simplest amino acids, and they're 5 of the earliest arriving amino acids on Earth, even before biological systems were available. 3 of the next 5 are in the next row up, and then leucine and proline are one row up from there, which then makes up 10 amino acids that were available on Earth to start out life on Earth. So something like this may have represented the initial genetic code, where a U would stand for valine, a C would stand for alanine, an A would stand for either aspartate or glutamate, and a G would stand for glycine. And so it may have been a one nucleotide genetic code at that point, and 4 or 5 amino acids only. So then this would have been a simple code. The first genetic code might have been one nucleotide stood for one amino acid in that P site for adding the amino acids together at the peptidyl transferase site. Next, a 2 nucleotide code may have existed, but some of the evidence shows that it may have gone directly to a 3 letter code almost immediately. And I won't go into all the evidence about that, but it seems more likely that you had a 3-letter code where the third position in the codon was quite variable, and that would then make the transition from a one-letter codon to a 3-letter codon much more likely. If you examine the most ancient proteins, those are the ones that are found in every living organism, or almost everyone, including bacteria, archaea, and eukaryotes. They've been around a long time, so they're in everything. Then you find that alanine, valine, glycine, and glutamate are the most frequently found, and aspartate is a little further down. But if you have a single genetic code, it would be A, G, U, and C. If there's a genetic code with 2 nucleotides, then it would be GA, GG, GU, GC. So you can see how that would be easy to convert to. And if it's a 3-letter code, usually the third letter, the third position, can be almost anything. And so that would be also easily accommodated in the evolution of the triplet codon. So now this is just showing how you could go to a 2-letter codon, 2 nucleotide codon, starting on that bottom row, quite easily going from GU, well, the original would be a one-letter codon, UCAG, a 2-letter codon, you just add a G, so GU, GC, GA, and GG to get those 4 or 5 amino acids in the genetic code. So again, you can then shift into either a 2 nucleotide codon or a 3 nucleotide codon, a triplet codon, which is what is present today, starting from the 1 nucleotide codon very, very early on. So the effect that would have then, going from a 1 nucleotide codon, you can only have 4 possible amino acids. You still can have some variation in what those amino acids are. These ones over here are nonpolar, and then moving over to the right where you have more polar groups. Moving to a 2 nucleotide codon, now there are 16 amino acids possible, including no stop codons. But then you can make a genetic code that looks somewhat like this, where it's mainly nonpolar on one side, more polar groups and reactive groups on the right-hand side. Then if you start building the genetic code up as you look at the frequencies of those amino acids and what was available early on Earth, then the next group that seems to have appeared are these ones, isoleucine and threonine. And again, these are 2 more of those that were available very early on Earth before any life appeared on Earth. You can follow the increases and decreases of these amino acids in this way too, so that all of the amino acids that have increased in frequency over the past 4 billion years are in green, and all of the amino acids in proteins that have decreased in the past 4 billion years are in red. And in general, the very simplest amino acids have decreased in frequency, and the more complex amino acids, that can only be produced biologically have increased in their frequencies in those proteins. So when all of the data is then analyzed, essentially you have the bottom row beginning, and then you have moving up on this left-hand side into isoleucine, threonine, leucine, and proline. And then some additions down here from glutamate, aspartate into glutamine and asparagine. And then over to histidine and arginine and serine down here. And then the final ones that were added are over here: tyrosine, cysteine, tryptophan, and also selenocysteine was added. Based on what we know now, the triplet code evolved very early, probably by 4 billion years ago. And then once that system with the ribosome and the mRNAs and tRNAs had been established with a triplet code, further evolution by adding additional amino acids occurred, and probably took over 1 billion years for that part of the evolution. And although there still is evolution going on in the translation system, the major portions were established probably by 3 billion years ago. So here's a few more details on that. Things may have started from alanine, although some studies have said they began from glycine. But anyway, either way, you get evolution of this initial group on the bottom. And again, this probably started from a single nucleotide acting as a codon, possibly a dinucleotide, and probably evolving into a triplet codon early on as well. But it's fairly certain that that initial nucleotide and now in the first position was a guanosine, and that probably stabilized that interaction between the codon and anticodon, because a GC pair is going to be one of the strongest pairs. And even if you have a U in the second position or an A in the second position, it'll still be held together because of the additional hydrogen bonding of the GC pair. Also note that the start codon was probably GU and probably GUN by that time. As I stated earlier, probably the next amino acids to be added are isoleucine and threonine, as well as arginine and proline, and this implies that you need at least an A in the first position and probably a C in the first position in some of the codons. It's been suggested that most of the other codons were stop codons that would halt translation at any of those points. And by this time there were 2 start codons, AUN, which encoded for isoleucine, and GUN that encoded valine. And the reason we think that is that those are start codons for certain proteins in some organisms. But by the time you have a genetic code this complex, then it's clear that it has to be a triplet codon. And so at this point, we're probably talking about a genetic code around 4 billion to 3.8 billion years ago. So here it is again, and now more of the genetic code needs to be filled in. And so there are more codons used, this time mainly for leucine. So now we have all 10 of those original amino acids and a genetic code that uses a triplet codon, an anticodon. Several codons can be the start in this case, so that can include any codon with U in the middle position. And again, we think that's true because in some organisms those are used as start codons for some of the mRNAs. The next thing that happens is additional amino acids are added to the genetic code, but these are amino acids that were not available early on Earth, and these are ones that can be formed by biological systems, including using the TCA cycle and acetyl-CoA and some other things to produce these. Also, an essentially universal start codon, AUG, was adopted, which encoded for a methionine. And then finally, some of the most complex amino acids that can only be formed biologically were added to the genetic code. And the number of stop codons was reduced, and the number of start codons was reduced also. So then that leaves us with the current genetic code, which is nearly universal. And in some cases, instead of having 20 amino acids possible, there are 21 amino acids, including selenocysteine. And some of your proteins actually use selenocysteine. Okay, so that's the evolution of the genetic code in kind of a nutshell. So that's the end of this video, and I will talk to you later.